A plant can rewrite the story of human history. Scattered across the globe are botanical puzzles, species thriving on continents separated by vast oceans long before any recorded voyage. This is not a matter of legend, but of pollen, seeds, and genetics. The central question is stark. Could the prehistoric distribution of certain plants be the forgotten evidence of ancient, unrecorded journeys across the Atlantic and Pacific? By the end of this exploration, you will have a clear framework to separate compelling evidence from debunked myth, understanding what these silent green witnesses might be telling us. We will examine how a humble tuber crossed the Pacific millennia before Captain Cook, and why the very DNA of cotton suggests a meeting of two worlds. This systematic investigation will reveal the strongest cases for pre-Columbian contact and confront the skeptical counterarguments. The stakes are a potential rewrite of our deepest past, suggesting human connections in an age we thought was defined by isolation. What does it mean for our world if the map of ancient human ambition was always far larger than we dared to believe? These botanical puzzles force us to start with a fundamental question. How do plants normally move across the world? Nature has its own ingenious, if often slow, delivery systems. The lightest pollen and tiniest seeds can be swept across continents by powerful winds. Tough, buoyant seeds like those of the coconut can survive a voyage of months on ocean currents, washing ashore on a distant beach to take root. Birds play a crucial role, consuming fruits and later depositing the undigested seeds far from the parent plant. And sometimes entire chunks of shoreline, tangled with soil and vegetation, break away in storms to become floating islands, carrying their cargo of life across the sea. These methods work beautifully for certain species, but they have clear and serious limitations. They are acts of random chance, not targeted delivery. A seed carried by a bird or a current lands where it lands. It must then find the exact right conditions to germinate, compete with native species, and survive long enough to reproduce. This is a tremendous long shot. The odds grow even longer when we consider plants with large, heavy seeds that cannot float or be carried by wind, or species that require very specific human intervention to thrive, like deliberate planting, pruning, or protection from pests. This brings us to the heart of the mystery, the concept of biogeographic anomalies. These are plant species found in two or more parts of the world, separated by vast oceans or impassable mountain ranges, with no continuous natural corridor between them. According to everything we know about natural dispersal, they should not be there. Their presence creates a jagged, illogical tear in the map of life. For some of these plants, the gap is so immense and the species so ill-suited to natural long-distance travel that the explanation of wind, water, or wings begins to feel insufficient. It is like finding a single, specific puzzle piece from one box perfectly placed inside another box on the other side of the house. The simplest explanation is not a miraculous gust of wind through a keyhole, but that someone carried it there. This is the threshold of the human-assisted dispersal hypothesis, the moment a botanical oddity becomes a potential artifact of forgotten human journey. One plant stands at the center of this debate, a humble root with a globe-spanning secret. The sweet potato, a staple food born in the Americas, presents one of history's most compelling botanical puzzles. Its story begins in the ancient soils of South and Central America, where it was domesticated thousands of years ago. Yet, centuries before any European explorer set foot in the Pacific, the sweet potato was already there. It was thriving across the islands of Polynesia, from Hawaii to New Zealand to Easter Island. This is not speculation. Archaeological digs have uncovered sweet potato remains in Polynesia, with radiocarbon dating placing them firmly in pre-Columbian times, some estimates suggesting as early as 1,000 years ago. The plant did not slowly island hop by natural means. It appeared fully formed as a cultivated crop in gardens an ocean away from its homeland. The most striking evidence, however, might be in the name itself. In the Quechua language of the Andes, the word for sweet potato is Kumara. Across the vast Pacific in parts of Polynesia, strikingly similar words are used like kumala and uamala. This linguistic echo, carried across thousands of miles of open sea, suggests the word traveled with the tuber itself. Now, consider the biology. The sweet potato is primarily propagated from cuttings or sprouts, not from seeds. Its seeds are tiny and not built for long sea voyages. The idea that a fragile vine fragment could survive a months-long journey on salty ocean currents, then wash up precisely on an island and be recognized, planted, and cultivated by people is a chain of miracles stretching credibility. The far simpler, more elegant explanation is human agency. Someone who knew the plant, who valued it as food, carried it deliberately on a voyage. This combination of archaeological proof, linguistic kinship, and biological improbability makes the sweet potato a cornerstone of the trans-Pacific contact theory. It is a quiet, enduring clue that the world's greatest ocean may not have been the barrier we once believed. But the sweet potato is not the only botanical traveler with a mysterious past. Another plant, perhaps even more ancient in its global journey, presents a puzzle that divides researchers. This is the bottle gourd, a humble vine whose hard-shelled fruit has been used by humans for thousands of years as a container, a musical instrument, and a cultural symbol. Its story is one of truly prehistoric proportions. Archaeologists have found its remains in sites across the Americas, Asia, and Africa, dating back over 10,000 years. This means the bottle gourd was already widespread across multiple continents at a time when, according to traditional history, those continents were completely isolated from one another. How did it get there? The initial answer from science seemed beautifully simple. The bottle gourd produces a fruit with a tough, waterproof rind. When dried, it is incredibly buoyant. A mature gourd could theoretically fall into a river, float out to sea, and drift across an ocean, its seeds protected inside, eventually washing ashore on a distant continent to sprout. This natural rafting hypothesis is perfectly plausible and could explain a global distribution without any human help at all. But then, genetics complicated the picture. DNA studies of ancient bottle gourd remains revealed something fascinating. There are two distinct genetic lineages. One lineage is found primarily in the Americas. The other is found in Asia and Africa. This suggests not one, but at least two separate domestications of the plant in different parts of the world from different wild ancestors. This genetic split is deep, hinting at a separation that occurred tens of thousands of years ago. So, did the gourds float across naturally, or did people carry them? The debate hinges on timing and context. If the gourds arrived by natural drift, they would have done so long before humans migrated into the Americas. They would simply be a wild plant waiting to be discovered and domesticated independently on different shores. However, some researchers point to the fact that these gourds appear very early in archaeological sites, often in contexts that suggest they were already being used by people. Could early human migrants have seen the value of this floating container and deliberately brought it with them on their journeys as a kind of pre-packed, multi-purpose tool? The bottle gourd sits at the crossroads of this debate. It is a perfect candidate for natural dispersal, yet its deep entanglement with human culture from our earliest days leaves room for doubt. It reminds us that in the deep past, the line between a natural object and a human tool could be incredibly thin, and a journey begun by ocean currents might have been completed by human hands. The genetic blueprint of a single plant can hold a map of forgotten voyages. This is the story written in the DNA of cotton, one of humanity's most ancient and important crops. Most of the world's cotton, the soft, fluffy fiber that clothes us, comes from a species known scientifically as Gossypium hirsutum, or upland cotton, domesticated in the Americas. But when scientists first peered into its genetic code, they found a profound and startling secret. This New World plant is not purely American at all. It is a living mosaic, a genetic fusion of two separate worlds. The technical term for this is an allotetraploid. In simpler terms, it is a hybrid that occurred when two different parent cotton species came together, each contributing a full set of chromosomes. One parent was a wild cotton species native to the Americas. The other parent, however, was a cotton species whose ancestors came from Africa or Asia. At some point in the deep past, these two plants, separated by an entire ocean, met and merged. Their genetic union created something entirely new, a stronger, more versatile cotton that would eventually be shaped by human hands into a global commodity. The critical question is, of course, how did that old world cotton ancestor reach the Americas? Molecular clock dating, a technique that estimates when genetic divergence occurred, places this hybridization event long before the voyages of Columbus, likely thousands of years earlier. This timing dismantles the simplest explanation. The genetic fusion was not a product of modern, recorded history. It was a prehistoric event. Now, consider the biology. Cotton seeds are not built for transoceanic travel. They are relatively large, they are killed by prolonged exposure to salt water, and they lack any obvious mechanism for long-distance dispersal by wind or birds. The odds of a viable seed from Africa or Asia surviving a months-long journey across the Atlantic, washing ashore precisely where a compatible American cotton species grew, and then successfully hybridizing in the wild, are astronomically small, a series of perfect accidents stacked one upon the other. This leaves a far more compelling, if controversial, implication. The genetic evidence whispers of a more deliberate journey. It suggests that the old world cotton ancestor did not drift aimlessly. It may have been carried, whether clinging to the hull of a vessel tucked among provisions or valued as a curiosity. Its movement across the ocean points toward human agency. The cotton genome, therefore, is more than a botanical record. It is a potential signature of contact, a silent witness to a moment when two separate branches of the world's flora, and perhaps the people who knew them, were brought together. Yet for every compelling case like cotton, there is a cautionary tale that shows how easily hope can outpace proof. This is the story of maize, a crop whose journey is meant to be one of history's clearest dividing lines. According to the established narrative, maize, what many call corn, was domesticated in Mexico over 9,000 years ago. It was the engine of great American civilizations, utterly unknown to the rest of the world until Spanish ships carried it across the Atlantic after 1492. Any suggestion of maize in the Old World before that date challenges a foundational pillar of agricultural history, and yet such suggestions have persistently surfaced. Over the decades, researchers and enthusiasts have pointed to curious artifacts. A faint, grassy pollen grain found in a sediment core from India, dated to centuries before Columbus. A sculpted stone figure from ancient Egypt that some argue resembles an ear of corn. A medieval European painting where a botanical detail looks, to some eyes, suspiciously like a maize plant. Each of these findings, when announced, sparks a flurry of excitement. Could they be the smoking gun for pre-Columbian contact across the Atlantic or the Pacific? The problem, and it is a profound one, is that none of these pieces of evidence have withstood rigorous scientific scrutiny. The pollen grain could be from a related wild grass native to Asia, its identification uncertain. The Egyptian sculpture almost certainly depicts a pine cone or a cluster of dates. its form reinterpreted through wishful thinking. The European painting is a case of mistaken identity, the plant in question being a more common Old World grain like sorghum or wheat. The overwhelming consensus from archaeology, botany, and history is resolute. There is no verified, uncontested evidence of maize in Asia, Africa, or Europe before 1492. Every claimed discovery has been explained by misidentification, contamination of samples, or the reinterpretation of local plants. This case is not a dead end, but a vital lesson. It highlights the immense importance of methodological rigor in this field. A single ambiguous data point is not enough to rewrite a chapter of history. It must be backed by clear, repeatable evidence, like the carbon-dated sweet potato remains in Polynesia or the genetic fingerprint in cotton. The Maize Controversy teaches us to temper excitement with extreme caution. It shows how the human desire to find connections can sometimes see patterns where none exist, turning common things into extraordinary proofs. In the search for lost histories, the discipline of saying, "we are not yet sure," is just as important as the thrill of discovery. Perhaps no plant embodies the romance of tropical shores more completely than the coconut palm. Its silhouette against a sunset is a universal symbol of island life, found on coastlines from the Caribbean to the Indian Ocean to the remotest Pacific atolls. This pantropical distribution presents its own elegant mystery. How did a single tree species come to dominate so many distant beaches? The classic and seemingly perfect answer lies in the fruit itself. The coconut is a masterpiece of natural engineering for ocean travel. Its thick, fibrous husk is buoyant and waterproof. Its hard inner shell protects the seed and its vital water reserve from salt. It can float for months, even years, surviving a voyage across entire oceans, finally washing ashore where the tide deposits it. Once there, the seed can germinate, sending roots into the sand just above the high tide line. This is a beautiful, self-contained dispersal story, and for many scientists it is a complete explanation. The coconut, they argue, simply rode the world's currents to its present home, a natural drifter long before humans arrived to appreciate its gifts. Yet the story grows more complex when we look closer, beyond the simple physics of floating. Recent genetic studies of coconut populations around the world reveal a fascinating pattern. The coconuts of the Pacific Basin are genetically distinct from those found in the Indian Ocean and the Atlantic coasts of the Americas. This suggests not one, but at least two separate origins or ancient lineages. More intriguingly, the genetic diversity of coconuts is highest in Southeast Asia and the surrounding islands, pointing to that region as a likely cradle of domestication. This is where the plot thickens. While a wild coconut might certainly float from Southeast Asia to, say, Polynesia, the coconuts that became integral to human cultures were not wild drifters. They were cultivated varieties, selectively bred over generations for larger size, sweeter water, and thicker meat. These domesticated traits would not have evolved on a random, deserted beach. They are the signature of human choice and care. This brings us to the critical question: did the coconut palm spread naturally first, and humans simply found it and improved it wherever it grew? Or did humans play an active primary role in carrying their preferred varieties across the seas, effectively planting the world's coastlines with a familiar, life-saving resource? The ecology of the tree offers a clue. Coconuts thrive in coastal zones, but they rarely penetrate far inland without human help. Their dense, localized groves on habitable islands often mirror patterns of human settlement more closely than patterns of ocean currents alone would predict. Furthermore, the coconut is deeply woven into the material and spiritual culture of Pacific peoples. It was not just a food source, but a provider of fiber, timber, and vessel. A plant so essential it likely would have been a high-priority cargo on any long, exploratory voyage. So, is the coconut a drifter or a stowaway? The most compelling answer may be both. Its original dispersal from an ancient homeland probably involved natural ocean currents, scattering a wild ancestor across a vast realm. But the coconut we know today, the one that defines islands and sustains communities, likely achieved its modern, global footprint with a great deal of human assistance. Prehistoric voyagers, recognizing its immense value, may have selectively carried the best nuts from island to island, accelerating and directing its spread far beyond what chance alone could accomplish. This case does not offer a simple verdict but a layered truth. It shows how the story of a single species can become entangled with our own, reminding us that in the ancient tropics, the line between a natural landscape and a human-tended garden was often beautifully and deliberately blurred. The fiery kick of a chili pepper seems to belong to every cuisine on Earth, yet the entire genus capsicum, from the mild bell pepper to the searing habanero, is a gift of the Americas, domesticated there thousands of years ago. This creates a tantalizing puzzle. There are historical and linguistic hints, particularly in India and Southeast Asia, that some chili varieties might have arrived before Christopher Columbus's voyages. Certain ancient texts, when later translated, use words for hot pepper that some researchers argue could refer to the New World chili, not the Old World black pepper. Similar debates swirl around linguistic roots and traditional names. The possibility is electrifying. Could a taste for heat have crossed an ocean long before recorded history? To answer that, we must scrutinize the nature of the evidence and the incredible velocity of change that followed 1492. The Columbian Exchange, the great swap of plants, animals, and diseases between the Old and New Worlds, was not a slow seep of ideas. It was a botanical explosion. When Spanish and Portuguese traders brought chili peppers to Asia and Africa, they encountered climates perfectly suited to the plant and cultures with a pre-existing love for pungent, spicy flavors. The adoption was instantaneous and revolutionary. Within decades, not centuries, chilies had woven themselves so deeply into local agriculture, cuisine, and even language that they felt ancient. They replaced or supplemented other spices, created entirely new dishes, and their names often borrowed from or confused with those of familiar local plants. This rapid, profound integration is the key to understanding the confusion. It creates a perfect illusion. When a plant becomes culturally fundamental in just a few generations, the line between ancient and newly ancient blurs beyond easy recognition. A recipe written in the 1600s that mentions chilies could be documenting a tradition that feels old but is, in the grand timeline, strikingly recent. The linguistic echoes might not be clues of prehistoric travel, but artifacts of this incredibly fast cultural adoption, where a new thing is described with the closest available old word. So, while the case of the chili pepper is less definitive than the sweet potato or cotton, it serves a crucial purpose in our investigation. It stands as a powerful warning about the shadows cast by the Columbian Exchange. The patterns it created of sudden and total integration can look eerily similar to the patterns we would expect from much earlier contact. It teaches us to be exceptionally careful, to demand evidence that can be firmly dated to before that world-altering year of 1492. In this search, the speed of change itself becomes a central character, showing how quickly a new flavor can make itself at home and how easily it can disguise its own surprising journey. Across the Atlantic, another tantalizing mystery unfolds, this time on the shores of Africa. If the sweet potato's journey to Polynesia challenges the Pacific narrative, could there be evidence of American crops taking root in Africa long before the age of Columbus? Some researchers point to a handful of intriguing, if controversial, clues. Early European explorers, arriving on the coasts of West Africa in the late 15th and early 16th centuries, occasionally reported seeing crops that sound, in their descriptions, remarkably like American plants. Accounts sometimes mention fields of a starchy tuber or a tall grain already under widespread cultivation by local communities. To proponents of pre-Columbian contact, these are not mere accidents. They are potential eyewitness records of a transfer that happened centuries earlier, suggesting that Atlantic crossings may have occurred in both directions, with currents or perhaps even vessels carrying more than just ideas. The most commonly cited candidate is once again the sweet potato. Some historical records from the Portuguese, for instance, note a batata or similar root being grown in regions like present-day Ghana or Nigeria not long after their first contact. If these were indeed the American sweet potato, already established as a staple, it could imply an arrival date predating European ships. Similarly, there are ambiguous references to a type of maize, though these are far more hotly contested. The argument hinges on the interpretation of these old texts and the reliability of the observers, who were encountering a completely new world of flora and often struggled to describe what they saw with precise botanical accuracy. Skeptics, however, offer a powerful and sobering counterargument. They suggest these early accounts are almost certainly misinterpretations. Africa has its own rich and ancient agricultural tradition with a suite of native crops that could easily be confused with American imports by an untrained European eye. The batata described could have been the African yam, a starchy tuber with a long history of domestication on the continent. The tall grain mistaken for maize might have been sorghum or pearl millet, fundamental African cereals that grow in similar conditions. The skeptical view insists that these explorer accounts reflect the initial confusion of cultural encounter, not proof of prehistoric transfer. They argue that the rapid and well-documented spread of American crops via Portuguese and Spanish trade routes after 1492 is more than sufficient to explain their presence in Africa by the time later explorers wrote their reports. This leaves the debate in a state of fascinating uncertainty. Unlike the genetic fingerprint in cotton or the carbon-dated remains in Polynesia, the African evidence is primarily textual and interpretative. It lives in the ambiguous space of historical description, where a single word can carry the weight of an entire theory. The ongoing scholarly discussion is less about proving contact and more about carefully sifting through layers of historical perception, separating wishful reading from genuine anomaly. It underscores a crucial lesson in this entire investigation. For a claim of pre-Columbian contact to move from intriguing possibility to accepted fact, it requires evidence that is not just suggestive but resilient, evidence that can withstand the scrutiny of multiple disciplines: archaeology, genetics, linguistics, and botany, all pointing in the same direction. The African mystery, for now, remains a compelling question mark on the map, a reminder that some of history's deepest secrets are still hidden in plain sight, waiting within the words of those who saw a world on the verge of catastrophic and beautiful change. Deep in the Amazon rainforest grows a tree whose sap would one day change the world. The para rubber tree, with its scientific name Hevea brasiliensis, is the primary source of natural latex. Its story is a powerful test case for our investigation. We must ask a very specific question. Is there any credible evidence that this remarkable substance, or the knowledge of how to use it, existed outside the Americas before the voyages of Columbus? The properties of raw latex are fascinating but initially impractical. When first tapped, it is a sticky, milky liquid. Left alone, it will slowly coagulate into a useless, brittle mass. The true magic and the reason for rubber's value lies in a complex chemical transformation discovered by indigenous Amazonian peoples thousands of years ago. They developed a process using the smoke from specific fires to vulcanize the latex long before the term was invented. This smoke treatment prevents decay and gives rubber its legendary elasticity, waterproof quality, and durability. This was not a simple, accidental discovery. It represented a profound, sophisticated understanding of material science, allowing for the creation of waterproof footwear, bouncing balls for ritual games, and flexible containers. This deep, specialized knowledge is the first critical point. For rubber to have been transferred to another continent, it wouldn't be enough for a seed or a cutting to somehow make the journey. The entire cultural and technical package would need to travel with it: the knowledge of which tree to tap, how to tap it without killing the tree, how to collect the latex, and the precise smoking process to make it stable and useful. The odds of that complete transfer happening by chance are vanishingly small. So, when we examine the historical record, we find a stark divide. There is no archaeological, textual, or artistic evidence for the use of natural rubber in Europe, Asia, or Africa before the 16th century. The first stunned reports from Spanish explorers describe seeing natives playing with balls that bounced magically, a substance utterly alien to the Old World experience. Rubber remained a curious novelty until the 19th century, when processes for its stable commercial production were independently developed. This creates a clarifying contrast in our search. The Para rubber tree highlights the crucial difference between the potential for transfer and actual evidence of transfer. A plant species might have the biological potential to be carried across an ocean. But without corroborating proof, carbon-dated remains, clear genetic markers of human dispersal, or unambiguous artifacts in a new context, the potential remains just that, an interesting possibility. The rubber tree story reinforces a vital principle. For a claim of pre-Columbian contact to hold weight, the evidence must extend beyond the mere presence of a plant. It should, ideally, point to the presence of the human knowledge that made that plant meaningful. The silence surrounding rubber outside the Americas before 1492 is, in its own way, as telling as the compelling whispers we hear from the sweet potato or the cotton genome. It reminds us that the map of ancient connection is not blank, but carefully and sparsely drawn, with each potential line requiring its own burden of proof. The pineapple's journey from a tropical American fruit to a global icon of luxury is a story written in art and appetite. This striking plant, with its spiky crown and sweet, segmented heart, was utterly unknown outside the Americas until Columbus encountered it on the island of Guadeloupe in 1493. He called it Piña de Indes, the pine of the Indians, noting its resemblance to a pine cone. Its arrival in Europe marked the beginning of a fascinating cultural transformation. Because the pineapple is perishable and difficult to cultivate in temperate climates, only the wealthiest could afford to import the fresh fruit or maintain the specialized hothouses called pineries needed to grow them. Owning, displaying, or serving a fresh pineapple became the ultimate demonstration of wealth and generosity, a direct symbol of one's ability to command the fruits of a newly connected world. This status solidified its image. Soon, the pineapple's form was carved into bedposts and gateposts, cast in silver for table centerpieces, and woven into fabrics, becoming a universal shorthand for welcome and extravagant hospitality. But this very speed of adoption creates a historical echo chamber, perfect for breeding myths of deeper antiquity. The question then becomes, in all the art and literature of Europe, Asia, or Africa before 1492, is there a single, credible reference to this unmistakable fruit? Enthusiasts have sometimes pointed to ancient Roman mosaics depicting pine cones or certain medieval carvings, suggesting the artists might have been depicting a pineapple. Yet every single one of these claims dissolves under scrutiny. The Roman pine cone, or pigna, was a common symbol of fertility and eternal life, its scaly pattern bearing only a superficial resemblance. Medieval herbal manuscripts sometimes included fantastical plants from travelers' tales, but none match the accurate botanical detail of a true pineapple. The hard truth is that all legitimate, identifiable representations of the pineapple in Old World art begin abruptly and precisely in the decades after Columbus's return. They appear first as curious specimens in botanical illustrations, then as prized objects in still life paintings, tracing the fruit's integration into the European imagination. There are no mysterious precursors. This clear, clean break in the artistic record is profoundly informative. Unlike the sweet potato, which appears in Polynesia centuries before European contact, the pineapple leaves no such shadow in the ancient Old World. Its story is a brilliant, self-contained capsule of the Columbian exchange. It demonstrates how a novel object can rocket from total obscurity to central symbolic status within a few human generations, its image spreading so fast and so completely that it feels like it must have always been there. The pineapple, therefore, teaches us to read the silence as clearly as we read the evidence. Its complete absence before a specific date is not a gap in our knowledge, but a confirmed boundary. It shows that some treasures of the New World truly were secrets kept by an ocean, waiting for a specific moment in history to unveil their spiked and golden glory to a waiting world. The story of tobacco introduces a detective story of the highest scientific stakes. This plant, with its potent alkaloid nicotine, was domesticated in the Americas and held profound cultural and ritual significance for countless indigenous societies. Yet whispers of a far older global footprint have emerged from an unlikely source, the resting places of ancient Egypt. For decades, reports have surfaced claiming that chemical analyses of Egyptian mummies, some dating back thousands of years before any possible contact with the New World, have revealed traces of nicotine and cocaine, another American plant product. If true, these findings would be the most explosive evidence yet for Trans-Oceanic contact, shattering timelines and rewriting trade routes in the ancient world. The methodology behind such claims involves highly sensitive chemical testing, like gas chromatography and mass spectrometry, which can detect minute molecular signatures in hair, bone, or tissue samples. The initial results, when announced, sent shockwaves through archaeology. They suggested a network of exchange so vast it could have connected the Nile with the Amazon. But extraordinary claims demand extraordinary proof, and the scientific process quickly turned its scrutiny on the evidence itself. The central problem became one of contamination and alternative sources. Nicotine, while most concentrated in tobacco, is not exclusive to it. It is found in smaller amounts in other plants from the nightshade family, which grow in the Old World, such as certain types of henbane or even some varieties of eggplant. More critically, the potential for modern contamination is immense. A researcher handling samples after smoking a cigarette, or even exposure to ambient smoke in a museum or laboratory over decades, could transfer microscopic nicotine residues to ancient remains. The preservation methods used in the 19th and 20th centuries, which sometimes involved chemicals or plant-based resins, add another layer of interpretive difficulty. Subsequent, more rigorously controlled studies have often failed to replicate the initial findings, or have offered plausible Old World botanical sources for the compounds detected. This does not necessarily close the case, but it shifts the burden of proof dramatically. It transforms the mystery from a question of history into a masterclass in modern scientific caution. The lesson of the tobacco and mummy controversy is not that pre-Columbian contact is impossible, but that proving it through fragile chemical signatures requires a chain of custody and analytical purity as pristine as the mummies themselves. It highlights the razor's edge between a world-altering discovery and a modern fingerprint inadvertently left upon the past. Another plant with a face turned toward the sun presents a different kind of puzzle, one of mistaken identity across the centuries. The sunflower, a towering bloom of golden petals radiating from a dark central disk, is unequivocally a child of the Americas. Its domestication began over 4,000 years ago in what is now the eastern United States, cultivated by indigenous societies for its nutritious seeds and oil. It was utterly unknown in Europe until Spanish explorers carried it back across the Atlantic in the 16th century. Yet a persistent idea suggests its image might have crossed the ocean far earlier, woven into the very fabric of ancient Greek myth. The logic seems enticingly poetic. The Greek sun god was Helios, who drove his fiery chariot across the sky. The sunflower's scientific name, Helianthus, literally means sunflower. Could the Greeks have known this American plant, naming their myths after its dazzling solar resemblance? Some point to ancient art and texts describing flowers that turn to follow the sun, seeing in them a clear reference to the sunflower's heliotropism. This would be a stunning piece of evidence, placing a New World species in the Old World imagination millennia before Columbus. To evaluate it, we must become botanical detectives, scrutinizing not just names but precise physical forms. The critical error here is a confusion of morphology. The domesticated sunflower, Helianthus annus, is a composite flower. What appears to be a single large bloom is actually a head or capitulum, made up of hundreds of tiny individual flowers. The outer yellow rays are sterile petals, while the dark brown central disc contains hundreds of tiny seed-producing florets. This architecture is unique and unmistakable. Now examine the plants of the ancient Mediterranean. Several species there exhibit sun-tracking behavior, a trait known as heliotropism. The common heliotrope, for instance, has small clusters of fragrant purple or white flowers. The pot marigold or calendula has a daisy-like form. Various chrysanthemums also display this behavior. None of them, however, look anything like the massive, singular-headed, seed-packed sunflower. When ancient Greek or Roman writers described a flower following the sun, they were almost certainly observing one of these native species. The word heliotrope itself comes from the Greek for sun turn. The conflation arises because we now use sunflower as the common name for the American Helianthus, grafting a poetic description onto a specific biological entity the ancients never saw. It is a case of linguistic coincidence layered over 2,000 years of translation, where a general behavior turning toward the sun has been retroactively attached to a specific, unrelated plant that perfectly embodies the metaphor. This chronological dilemma dissolves under straightforward botanical scrutiny. There are no accurate depictions of the true sunflower in European art or literature before the age of exploration. The myth of Helios was inspired by the celestial body itself, not by a flower that would remain a secret of another continent for ages to come. This case underscores a vital rule in the search for pre-Columbian contact. We must guard against the powerful allure of symbolic resonance. A name that fits perfectly, a behavior that mirrors a myth. These are compelling narratives. But without the precise material evidence, the correct seed in the right archaeological layer, the accurate image in the ancient text, they remain just that, stories. The sunflower's true story is magnificent enough, a journey from ancient American gardens to global fields, its face forever turned not toward a forgotten Greek past, but toward the open sky of a connected world. The protein that fueled ancient American civilizations came from a humble source, the common bean. This is the genus Phaseolus, a family that includes the kidney bean, the pinto, the black bean, and the lima bean, all domesticated thousands of years ago in the New World. Their story intertwines with maize and squash in the legendary Three Sisters agricultural system, a cornerstone of life from Mexico to the Andes. So when we ask if beans might have crossed an ocean before Columbus, we are asking a very specific botanical question. Are any of the beans eaten in Europe, Asia, or Africa before the 1500s actually American Phaseolus species? The answer requires a dive into the science of classification, because the word bean is a culinary term, not a scientific one. It is a label we give to many large, edible seeds, and that creates immense confusion. In the ancient Old World, people had their own beloved pulses. The fava bean, a staple of Mediterranean and Asian diets for millennia, is not a phaseolus at all. Its scientific name is Vicia faba, placing it in an entirely different genus with a separate evolutionary history. The same is true for the chickpea, the lentil, the pea, and the mung bean. Each belongs to a different botanical group native to the Old World. This taxonomic distinction is not just academic trivia, it is the bedrock of the debate. If someone claims evidence of beans in ancient Egypt or Rome, the first question must be, which bean? Archaeologists find seeds all the time. The critical work is identifying them correctly down to the genus or species level. To date, no archaeological site in Europe, Africa, or Asia has yielded a securely identified pre-Columbian seed of Phaseolus vulgaris or any of its American cousins. Not one. All confirmed archaeological finds of true Phaseolus beans outside the Americas appear in layers dated after the 1500s, following the now familiar pattern of the Columbian exchange. Genetic studies reinforce this wall of separation. The DNA of American Phaseolus species shows no deep, prehistoric kinship with Old World pulses like the fava. Their lineages diverged on different continents tens of millions of years ago, long before any possibility of human-mediated contact. They evolved in parallel to fill a similar ecological and culinary niche, a beautiful example of convergent evolution where two separate branches of life arrived at a similar nutritious solution. So the common bean presents a case of remarkable clarity. Unlike the sweet potato with its linguistic echoes in Polynesia, or cotton with its hybrid genome, the bean offers no ambiguous middle ground. The evidence is consistent and unanimous. All Old World beans before 1492 were their own native varieties, and all New World beans were confined to their hemisphere. This clarity serves a crucial purpose in our investigation. It establishes a firm baseline, a control group. It shows that the absence of evidence can, in some cases, be evidence of absence. It tells us that the global pantry was indeed compartmentalized, that entire categories of staple foods remained profound secrets of their continents. The fact that some plants, like the sweet potato, seem to break this rule makes their cases all the more exceptional and demands that we scrutinize them even more carefully. The humble bean, therefore, is not a disappointment in the search for lost contact. It is a guidepost. It defines the normal boundaries of the ancient world, so that the true anomalies, when we find them, shine all the brighter against a map we thought we understood. Its peculiar biology offers a unique test for our theories. The peanut does not behave like other plants. After its small yellow flowers are pollinated above ground, something remarkable happens. The flower stalk, called a peg, begins to bend downward and push into the soil. Only underground, in darkness, does the actual peanut pod develop and mature. This geocarpy, or earth fruiting, is a brilliant adaptation for surviving dry conditions, but it creates an immense barrier for accidental travel. Consider the logistics of natural dispersal. A peanut seed snug in its pod is buried. For it to move any significant distance, the entire pod would need to be unearthed by an animal and carried away, only to be buried again in a suitable spot. The idea of that pod then surviving a months-long saltwater journey, washing ashore on another continent, and being buried at the perfect depth by pure chance is a sequence of events so improbable it borders on the fantastical. Unlike a floating coconut or a wind-borne seed, the peanut is an earthbound anchor, biologically chained to the very spot where its flower chose to dig in. This makes it an exceptionally clear marker. If we were to find evidence of the peanut in the Old World before 1492, it would almost certainly point to deliberate human transport. Someone would have had to carefully dig up the pods, recognize them as a valuable food source, preserve them during a voyage, and deliberately plant them in a new land. So, has such evidence ever surfaced? Over the years, a few speculative claims have emerged, often pointing to ambiguous artistic depictions or questionable interpretations of ancient texts. A carving in a temple might be said to resemble a peanut plant, or a phrase in an old manuscript might be translated as ground nut. Yet, just as with maize and the sunflower, these claims dissolve under focused scrutiny. The carvings depict other, native fruits or symbolic motifs. The textual references are conclusively to Old World legumes like the tiger nut or certain types of peas. Archaeologically, the record is silent and definitive. No peanut shells, no pods, no pollen of the Arachis genus have ever been recovered from a securely dated, pre-Columbian site in Europe, Africa, or Asia. Its documented history outside the Americas begins with the Spanish and Portuguese, who quickly spread it to Africa and Asia where it flourished. The peanut story, therefore, becomes a powerful testament to isolation. Its stubborn, subterranean nature makes it a near-perfect control in this grand experiment. While the sweet potato's journey whispers of possible connection, the peanut's steadfast absence shouts of a world still divided. It reinforces a crucial, grounding principle. The fact that some plants may have crossed oceans only makes the plants that did not cross all the more significant. They remind us that these ancient worlds, for all their potential hidden threads, remained largely separate gardens, each cultivating its own unique treasures in splendid and prolonged isolation until the moment the map was finally, irrevocably, stitched together. A final taste, perhaps the most luxurious of all, brings us to the sacred bean of the Americas, the cacao tree. Theobroma cacao, literally meaning food of the gods, was far more than a crop in ancient Mesoamerica. It was a form of currency, a ritual beverage, and a cornerstone of social and economic life for cultures like the Maya and Aztec. Its beans were so valued they were used as money, a fact that stunned European arrivals. This deep cultural entanglement presents a sharp question. Given its immense importance, is there any whisper, any fragment of credible proof that chocolate or the knowledge of cacao existed in Europe, Asia, or Africa before Spanish galleons carried it across the Atlantic in the 16th century? To answer, we must understand cacao's profound secret. The raw cacao bean, as it comes from the pod, is intensely bitter and inedible. The transformation into something recognizable as chocolate is not simple. It requires a precise, multi-step process of fermentation, drying, roasting, and grinding, a technology developed and perfected over centuries in the Americas. The beans must first be fermented in their own pulp for several days, a crucial step that develops the complex flavor precursors and kills the seed's germination ability. This is not an intuitive process one discovers by accident. It is a sophisticated cultural invention. So, for cacao to have appeared in the Old World before recorded contact, it would not have been enough for a single pod to somehow drift across the sea. The entire intricate knowledge system, the understanding of which tree to use, when to harvest, how to ferment, dry, and process the beans would have needed to travel intact. There is no archaeological trace of this in the ancient Old World. No cacao residue on pottery shards from ancient Egypt or Rome. No artistic depictions of the distinctive, rugby ball-shaped pods adorning temples in India or Southeast Asia. No written records in Greek, Arabic, or Chinese texts describing a bitter bean used as money or whipped into a frothy, spiced drink. The technology to process it simply did not exist outside its native hemisphere. The absence is total and telling. Cacao's story, like that of rubber, reinforces a critical distinction in this search. Some plants might be transferred as simple curiosities. But a plant woven so completely into the economic and spiritual fabric of a society represents a much heavier cultural package. its journey would leave a much louder echo. The silence surrounding cacao outside the Americas before the 1500s is not a mystery to be solved, but a boundary confirmed. It tells us that some treasures were so deeply rooted in their own world that they remained its exclusive secret, waiting to be unveiled and then, inevitably, transformed into a global obsession. A final flavor from the New World completes this culinary puzzle. The tomato, Solanum lycopersicum, began its journey high in the Andes, where it was first cultivated by indigenous peoples long before it ever saw a pasta sauce. Its path to global dominance is a classic story of the Columbian exchange, arriving in Europe in the 16th century and slowly overcoming initial suspicion to become a kitchen staple. Yet this very familiarity has spawned a persistent set of myths, a desire to see the tomato's red fruit peeking out from the corners of history long before it was possible. Across the internet and in some popular histories, you will find claims that the ancient Romans or medieval Europeans knew the tomato. The evidence presented is almost always visual, a piece of artwork where a round, red object in a painting or a mosaic is declared to be a tomato. One common culprit is the mandrake root, a plant with a thick, often forked shape that was believed to have magical properties and was frequently depicted in medieval herbals. To a modern eye, a stylized painting of a mandrake might vaguely resemble a cluster of tomatoes. Another is the pomegranate, a fruit deeply symbolic in both classical and religious art, its bursting red seeds easily mistaken for the smooth skin of a tomato in a centuries-old illustration. These misidentifications are understandable at a glance, but they crumble under the discipline of iconographic analysis. This is the method art historians use to decode the meaning of symbols within their specific cultural context. An expert looking at a medieval manuscript doesn't just see a red circle. They ask what that symbol meant to the artist and the audience of the time. Is the plant shown alongside other known Old World species? Is it labeled with a name that corresponds to a documented plant of the era, like Mandragora or Malum granatum, the pomegranate? Does its depiction match the artistic conventions for that plant, not our modern photographic expectation? In every single alleged case of a pre-Columbian tomato in European art, the answer is no. The artistic record is unanimous and clear. All accurate, identifiable depictions of the tomato begin abruptly in the late 16th century, first as a curious New World specimen in botanical catalogues, often called a love apple or golden apple, and only later as a food item. This case is a masterclass in separating wishful thinking from material proof. The tomato's story shows us that the tools for investigating these mysteries are not held by botanists alone. The art historian, with their understanding of symbolism and cultural context, is just as crucial a detective. They help us read the visual language of the past, protecting the timeline from the powerful allure of a pattern that simply isn't there. It is a reminder that sometimes the most important evidence for what wasn't there comes from carefully learning to see what actually was. The story of our global garden grows even more complex with a family of plants that provided some of humanity's first containers and most reliable food stores. This is the Cucurbitaceae family, a sprawling clan that includes gourds, melons, and squashes. Within this family lies a clear botanical divide, a harvest of questions that helps map the ancient world. On one side are the classic Old World cucurbits, plants like melons and cucumbers, domesticated in Africa and Asia and familiar to ancient Egyptian, Greek, and Roman tables. On the other side stand the great New World domesticates, the true pumpkins and winter squashes, species like Cucurbita pepo, which gives us the classic field pumpkin and acorn squash, and Cucurbita machata, the source of the butternut squash. These were not just food, they were technological innovations. Their hard shells made perfect, durable containers and vessels long before pottery was widespread, and their dense, storable flesh provided a vital calorie reserve through lean seasons. The archaeological record in the Americas tells a story of astonishing antiquity. Remains of domesticated cucurbita have been found in Mexican caves dating back over 10,000 years, making them some of the very first plants humans ever brought under cultivation anywhere on Earth. These ancient seeds and rind fragments are milestones in the human journey, marking a deliberate turn from foraging to farming. Now, shift the focus across the oceans. Despite decades of meticulous excavation, not a single seed, not a fragment of rind from a New World cucurbita species has ever been confirmed in a pre-Columbian archaeological site in Europe, Africa, or Asia. The silence is absolute. Old World sites yield plenty of melon seeds, grape pips, and fig remains, but the distinctive seeds of the pumpkin or the tough shell of a butternut squash are entirely absent. This negative evidence is, in its own way, powerfully positive. It reinforces the New World provenance of these essential crops with a clarity that few other lines of evidence can match. It tells us that while human societies on different continents both discovered the usefulness of hard-shelled, vining plants, they worked with an entirely separate botanical toolkit. One hemisphere cultivated its melons and cucumbers, the other perfected its pumpkins and squashes, each in profound and prolonged isolation, developing their own unique culinary traditions around fruits that would one day, but not yet, become neighbors in the same autumn field. The evidence we have explored presents a fascinating mosaic, but it is far from a settled map. To see the full picture, we must also listen to the powerful, prevailing voice of mainstream science, which offers a different explanation for these botanical puzzles. This is the principle of convergent evolution and independent domestication. It suggests that humanity's story is not one of unexpected connections across vast oceans, but of parallel ingenuity on separate continents. When faced with the same fundamental challenges, the need for reliable starch, durable fiber, or storable calories, different human societies, working with the wild plants available in their own environments, often arrived at strikingly similar solutions. They looked at their local flora and, through generations of selective breeding, shaped different wild ancestors into plants that served the same purpose. This process can create what scholars call a profound illusion of relatedness. Two plants on opposite sides of the world can end up looking and functioning so similarly that they appear to be close relatives, suggesting one must have traveled from a single point of origin. Yet their genetic blueprints tell a different story, revealing they evolved from completely separate wild stock. Consider the most basic human staples. In the Fertile Crescent, ancient peoples domesticated wild grasses like emmer and einkorn, which became wheat. In East Asia, a different set of wild grasses was shaped into rice. And in Mesoamerica, yet another wild grass, teosinte, was transformed through millennia of human selection into maize. All are grasses, all provide carbohydrate-rich grains, but each came from a distinct wild progenitor in its own hemisphere. No one argues that wheat was carried to China or that rice was brought to Mexico in prehistory. We accept that these parallel domestications happened independently because the archaeological and genetic evidence is overwhelming. This framework asks us to apply the same rigorous logic to every curious case. Could the sweet potato in Polynesia be the exception that proves the rule, or does it, too, have a wild ancestor somewhere in the Pacific that we simply have not yet identified? Could the similarities in certain plants be a testament not to human voyages, but to the universal nature of human need and botanical potential? This scientific perspective urges profound caution. It reminds us that correlation is not causation, and that a plant found in two places is not automatically evidence of a journey. It might instead be evidence of two separate journeys of discovery. two independent moments where human curiosity and patience coaxed a wild species into a trusted partner. Before we draw lines across oceans on the map of prehistory, we must first exhaustively rule out the possibility that we are simply seeing the same brilliant human idea, written twice in two different botanical languages. These botanical mysteries are no longer left to speculation alone. Modern science has brought powerful new tools to the silent testimony of seeds and pollen, transforming whispers into data. The field of Paleobotany, the study of ancient plants, now works hand in hand with archaeogenetics, a discipline that deciphers the DNA of the past. Together, they provide a forensic toolkit for investigating prehistory, offering answers that written records never could. The first crucial tool is radiocarbon dating. This technique measures the decay of a radioactive isotope in organic material, allowing scientists to determine with remarkable precision when a plant lived. A charred seed, a fragment of rind, or even a piece of ancient basketry can be dated. This is how we know beyond doubt that sweet potato remains in the Cook Islands are over 1,000 years old. It provides the irrefutable timestamp, placing the plant in a location long before any European ship could have brought it. But dating only tells us when, not necessarily how it got there. This is where ancient DNA analysis enters the story. By extracting and sequencing genetic material from archaeological plant remains, scientists can trace a specimen's lineage. They can identify its species with certainty, distinguishing, for example, an American cucurbita squash seed from an Old World melon seed in a way that visual inspection alone never could. More powerfully, they can reconstruct family trees. They can show that the cotton grown in Peru 2,000 years ago carries that signature hybrid genome, proving the genetic fusion was not a modern event. Ancient DNA turns a simple seed into a historical document, one that records its own ancestry and the moment it met another branch of its family across an ocean. Then there are the silent, nearly indestructible witnesses, phytoliths. These are microscopic particles of silica that form within the cells of many plants, taking on distinctive shapes unique to each species. When the plant decays, these tiny, glass-like skeletons persist in the soil for millennia. A researcher can take a soil sample from an ancient hearth or a storage pit, process it, and under a microscope, identify the ghostly imprint of plants that left no other trace. Phytoliths have revealed the presence of maize in areas where no kernels were preserved, and they can tell us what people were growing, eating, or even weaving, long after every other part of the plant has vanished. The power of this combined approach is its objectivity. It does not rely on interpreting ambiguous art or translating uncertain words. It deals in chemical signatures, genetic codes, and physical forms that are either present or absent. This scientific rigor acts as a filter, separating compelling evidence from wishful thinking. It has confirmed the profound anomaly of the Polynesian sweet potato, giving weight to the theory of human-mediated dispersal. At the same time, it has systematically debunked countless other claims. Those reports of ancient Egyptian maize, not a single one has produced a radiocarbon-dated cob or a definitive maize phytolith from a secure pre-Columbian context. The alleged tobacco in mummies, repeated, more stringent DNA and chemical analyses have failed to confirm it, pointing instead to contamination or other botanical sources. These tools do not automatically support any single narrative. They serve the truth of the material itself. They remind us that the past is not a story we get to write, but a puzzle we must carefully, patiently, and humbly learn to read, one microscopic fragment and one genetic letter at a time. Drawing these threads together, we can trace a faint but compelling line across the greatest ocean on Earth. It is not a route found on any ancient map, but one written in the dispersal of plants and animals. Scholars sometimes call this the Kumara line, a hypothetical pathway of contact forged by the most accomplished open ocean navigators in human history, the Polynesians. This scenario synthesizes the strongest evidence into a coherent story. It begins with the sweet potato, that American tuber with its Polynesian name and pre-Columbian presence. It incorporates the bottle gourd, whose early Pacific presence could be natural or assisted, and it is bolstered by a fascinating genetic clue from an animal, the chicken. Ancient chicken bones found in Chile have been linked through DNA analysis to prehistoric poultry from Polynesia, not to European breeds. While this finding has been debated, it adds another potential strand to the tapestry. The proposed voyage follows the logic of exploration. Polynesian voyagers, masters of reading stars, waves, and bird flight, push eastward across the Pacific. They eventually make landfall on the coast of South America. There, they encounter new people and new resources. Among the most valuable is the sweet potato, a hardy, nutritious root crop perfectly suited for the long voyage home. They collect cuttings, and on the return journey, they carry this new treasure westward, introducing it to their island homes. This was not a colonization, but an exchange, a moment of connection that left a botanical signature across thousands of miles. The navigational feasibility of such a journey is no longer in question. We know from experimental voyages like that of the Hokulea that traditional Polynesian double-hulled canoes, using non-instrumental wayfinding, are fully capable of such long-distance, two-way travel. The growing, though not yet conclusive, archaeological support adds weight. Beyond the chicken bones, there are tantalizing hints, like possible Polynesian-style fishhooks found in Chile and similarities in certain tool technologies. The Kumara line remains a hypothesis, but it is a powerful one. It provides the simplest explanation for a cluster of evidence that otherwise floats in isolation. It suggests the Pacific was not a void, but a highway, traversed by explorers whose legacy grows not in stone monuments, but in the quiet roots of a humble, sustaining vine. While the Pacific offers a compelling story, the Atlantic presents a colder, murkier mystery. This vast and stormy ocean seems a far more formidable barrier, its currents less forgiving, its distances just as great. Yet here too, whispers of forgotten crossings persist, clinging to the edges of history like sea mist on a rocky shore. We have one solid, undeniable data point, a confirmed landing on the far side. This is the Norse settlement at Elance Orks Meadows in Newfoundland, carbon dated to around the year 1000. These were the Vikings, master shipbuilders and navigators of the North Atlantic, who hopped from Scandinavia to Iceland to Greenland, and finally, to the very fringe of the American continent. Their presence proves that Atlantic crossing was possible with early medieval technology, but it was also fleeting, a small outpost that did not lead to lasting colonization or, as far as we know, a significant exchange of crops. It serves as a crucial precedent, a beacon in the fog that shows us where one path definitively led. But it also raises a haunting question. If they could do it at the turn of the last millennium, could someone else have done it even earlier? The search for pre-Viking contact across the Atlantic is where the evidence grows thin and the speculation grows thick. Unlike the Pacific, with its sweet potato and chicken dinner, the Atlantic lacks a single, universally accepted botanical smoking gun. The claims that do exist often focus on the opposite direction of travel, suggesting Old World plants found their way to the Americas long before Columbus. You might hear of alleged evidence for ancient Roman coins in New England, or stories of Celtic ogham inscriptions carved into stones in the American Southeast. These artifacts are almost universally dismissed by mainstream archaeologists as hoaxes, misidentifications, or objects lost by much later collectors. The botanical evidence in this direction faces the same intense scrutiny. There have been claims, for instance, that the bottle gourd in the Americas arrived from Africa, or that a type of Old World cotton somehow made it over. But as we have seen, the genetics of these plants often tell a more complex story of ancient, natural dispersal or independent domestication. The Atlantic currents, particularly the powerful Gulf Stream, do theoretically allow for drift voyages from Europe to the Caribbean or North America. The idea is not impossible. A disabled vessel or even a deliberate expedition could have been carried across. The problem is not feasibility but proof. Where is the material evidence? Where are the pre-Columbian settlements with distinctly Old World tools, the burials with genetic markers unambiguously pointing to Europe or Africa, the cache of seeds from a Mediterranean plant found in a securely dated American archaeological layer? That evidence on a scale needed to rewrite history simply has not materialized. The Atlantic, for all its rumors, guards its secrets more closely than the Pacific. Its potential pre-Viking contacts remain in the realm of intriguing possibility rather than demonstrated fact, a reminder that in the search for lost worlds, an ocean of doubt can be just as wide and deep as the sea itself. Yet for every thread of evidence there is a counter-thread of doubt, a necessary skepticism that keeps the search honest. The Trans-Oceanic contact hypothesis, for all its compelling mysteries, faces rigorous and legitimate critiques from the scientific mainstream. The first and most powerful argument is the underestimated resilience of nature itself. Proponents of natural dispersal remind us that the world is ancient, and chance events, however improbable, are not impossible over spans of thousands or even millions of years. A single successful voyage by a floating seed every 100,000 years is enough to establish a species on a new shore. Over deep time, the unlikely becomes inevitable. This perspective asks us to consider whether we are underestimating the creative chaos of wind, water, and wings, imposing a human narrative of deliberate voyage onto what is simply the slow, stately dance of biogeography. Another critical point concerns the integrity of the archaeological record itself. The layer of soil that holds an ancient seed is not always a sealed vault. Rodents burrow, roots penetrate, and later human activity can churn the earth. A sweet potato tuber found in a Polynesian midden might be carbon dated to 1,000 years ago, but could it have worked its way down from a later layer, perhaps from a garden planted after European arrival? This process, called stratigraphic intrusion, is a constant concern for archaeologists, proving that a specimen was found in a truly undisturbed, sealed context is paramount, and for some controversial finds that certainty has been challenged. Similarly, the specter of modern contamination haunts sensitive chemical analyses, like those seeking nicotine in mummies. A museum specimen handled by a smoker in the 19th century, or stored in a cabinet treated with certain preservatives, could easily acquire traces of modern compounds, creating a false signal of ancient presence. Even the most trusted tool, radiocarbon dating, is not infallible. The method relies on measuring the decay of a specific isotope, but that signal can be distorted. If an ancient seed absorbs more recent carbon from its environment, perhaps from groundwater, the dating can return a younger age than the object truly is. Conversely, if it comes from a part of a plant that took in old carbon, like deep roots, it might appear centuries older. Researchers must account for these reservoir effects, and in some high-profile cases, redating of specimens with improved techniques has shifted timelines, turning a potential anomaly into a mundane coincidence. Perhaps the most profound critique is not about data, but about human psychology. We are, by nature, storytellers. We are drawn to narratives of connection, of heroic voyages and lost civilizations, far more than we are drawn to the slow, incremental processes of independent innovation and random dispersal. A theory of ancient trans-oceanic contact is fundamentally romantic. It speaks to a deep-seated desire for a more interconnected ancient world, one where human curiosity and courage bridged the great blue voids long before the history books say it was possible. The alternative explanation that great civilizations rose in complete isolation, inventing agriculture, pyramids, and astronomy separately, can feel somehow less satisfying, a tale of parallel solitudes. This psychological bias is a powerful trap. It can lead us to cling to a single piece of ambiguous evidence while dismissing a mountain of contradictory data, or to interpret a vague artistic motif as a precise botanical portrait. Good science requires guarding against this allure, this preference for the wondrous story over the simpler, often more probable explanation. This skepticism is not cynicism. It is the essential immune system of historical inquiry, challenging each new claim to prove itself beyond a reasonable doubt. It ensures that if we one day do redraw the map of the ancient world, we will do so not on the basis of wishful thinking, but on a foundation of evidence so solid it can change the minds of even the most ardent skeptics. This debate is far more than a quarrel over seeds and sailing routes. It strikes at the heart of how we understand human history itself. For over a century, two opposing schools of thought have framed this conversation, creating a pendulum that swings between radical connection and total isolation. On one extreme lies the idea of hyper-diffusionism. This is the belief that all major cultural advances, from pyramids to agriculture, radiated out from a single, often lost, cradle of civilization. In its most dramatic forms, it suggests that a sophisticated seafaring people, perhaps from Atlantis or ancient Egypt, traveled the globe, bestowing the gifts of culture upon less developed societies. This theory, popular in the 19th and early 20th centuries, often relied on superficial similarities, like the presence of pyramid-shaped structures in both Egypt and Mesoamerica, to argue for direct contact and inheritance. It is a seductive narrative offering a simple, linear story of human progress. By the mid-20th century, however, the pendulum had swung hard in the opposite direction. A new, isolationist model took firm hold in academia. Reacting against the speculative excesses of hyper-diffusionism, scholars emphasized the incredible power of independent invention. They pointed to the overwhelming archaeological evidence showing that agriculture was domesticated in at least 7 separate regions of the world at different times, using completely different plants. They highlighted how monumental architecture, writing systems, and complex societies arose independently in Mesopotamia, the Indus Valley, China, Mesoamerica, and the Andes. This perspective celebrated human ingenuity, arguing that when faced with similar environmental and social challenges, different groups of people, completely cut off from one another, would arrive at remarkably similar solutions. For decades, this became the dominant and rigorously defended paradigm. To suggest trans-oceanic contact before Columbus was often seen as fringe thinking, a step back toward unscientific myth-making. The botanical evidence we have been examining, however, does not fit neatly into either of these rigid boxes. It suggests a third way, a more nuanced and fascinating middle ground. The sweet potato in Polynesia is not evidence of a lost civilization gifting culture to the Pacific. It is evidence of a specific, tangible exchange. The hybrid genome of cotton does not mean Old World farmers colonized the Americas. It signals a singular, ancient biological meeting. These are not the signatures of a global cultural blueprint being imposed, but of occasional, remarkable, and likely sporadic episodes of long-distance contact. Think of it not as a continuous bridge between continents, but as the rare and fleeting spark of a signal flare seen across a vast, dark ocean. Such contacts may have introduced a new crop, a novel technology, or a fresh idea, but they did not found civilizations. The Maya, the Inca, the Khmer, the Egyptians, these were profoundly original cultural creations, born from their own unique environments and histories. The possibility of a Polynesian voyage to South America does not diminish the astounding navigational achievements of the Pacific Islanders, nor the independent cultural brilliance of Andean societies. It simply suggests that at some point their stories briefly touched. This middle ground asks us to hold two powerful truths in balance. The first is the overwhelming reality of independent innovation, the incredible capacity of human societies to invent their own worlds in isolation. The second is the enduring human spirit of exploration, the drive to push beyond the horizon, which sometimes, against all odds, led to a meeting that left a seed, a word, or a gene as its only lasting trace. The story of our past, therefore, is not a choice between complete connection or total solitude. It is likely a mosaic of long, quiet periods of isolated development, punctuated by rare, luminous moments of unexpected connection, whose faint echoes we are only now beginning to hear. So how do we weigh this garden of conflicting clues? Let us sort the evidence into a clear framework, separating the robust from the speculative. At the top tier, we find cases built on multiple, converging lines of proof. The sweet potato in Polynesia is the prime example. It has the carbon-dated archaeological remains, the linguistic kinship in its name, and a biology ill-suited to natural drift. This is not a single data point, but a constellation of evidence that strongly points to human transfer. It stands in Tier 1. In a second tier, we place the fascinating puzzles where the evidence is compelling, but the interpretation remains actively debated. The bottle gourd fits here, its global spread plausible by natural means, yet its very early appearance in human contexts leaves a lingering question. The hybrid genome of cotton belongs here as well, a genetic signature of a meeting between two worlds that is difficult to explain without some form of biological transfer. Yet the precise mechanism of that transfer, natural or human-assisted, is still parsed by geneticists. These are not proofs but powerful anomalies that demand explanation. The final tier holds the cases that, despite persistent myths, have been systematically addressed by science. The idea of maize in ancient Egypt, sunflowers in Greek art, or tomatoes in medieval Europe falls squarely here. Each has been examined and, upon rigorous scrutiny, attributed to misidentification, contamination, or the powerful illusion of symbolic resemblance. They are lessons in caution, reminders of how easily we can project our modern world onto the past. When we step back and look at this tiered assessment, a nuanced picture emerges. No single plant, not even the mighty sweet potato, can definitively rewrite the history books on its own. History is rarely overturned by one artifact. But the cumulative weight of the evidence, particularly from Tier 1 and Tier 2, becomes difficult to dismiss. It suggests that the ancient oceans were not absolute walls. They were formidable barriers, yes, but barriers that were occasionally, remarkably, breached. The story these plants tell is not of a globally connected ancient world, but of a world where long periods of profound isolation were punctuated by rare, extraordinary moments of connection. A Polynesian canoe reaches a South American shore. A seed, cherished or accidental, survives a voyage. These were not colonizing missions or sustained trade routes, but fleeting sparks of contact that left their quiet, living signature in the soil and in the very DNA of the crops we still grow today. The map of prehistory, therefore, may need a few faint, tentative lines drawn across the blue, not as highways, but as testaments to those rare journeys when human curiosity briefly triumphed over the immense, isolating sea. The quiet evidence of a single plant can feel like a tremor beneath the foundations of history. But what if that tremor does not bring the entire structure down, but instead reveals a hidden room we never knew existed? The verified reality of pre-Columbian contact would not rewrite the main narrative of human civilization, but it would add breathtaking footnotes to its margins. It would not diminish the towering, independent achievements of the Maya, the Inca, or the great cultures of the Mississippi. Their pyramids, their calendars, their vast road networks and complex societies were not built from old-world blueprints. They emerged from the unique soil of their own continents, shaped by local genius and necessity over thousands of years. A sweet potato arriving on a Polynesian canoe does not build a city at Teotihuacan. The genetic fusion in a cotton plant does not chart the movements of Venus. These civilizations stand as monumental testaments to the human capacity for innovation in profound isolation. The real story that contact would tell is a different one, a story of parallel brilliance occasionally brushing against itself. It would elevate our understanding of ancient mariners, not as mythic figures, but as practical, daring explorers. To conceive of a voyage across the trackless Pacific, to build vessels capable of surviving it, to navigate by stars and waves and the flight patterns of birds, and to return home. This represents a staggering feat of technology and courage, equal in its own way to the engineering of a pyramid. It suggests that the human drive to explore, to see what lies beyond the horizon, is even more ancient and widespread than we credited. Our textbook timelines often present a neat, compartmentalized world where continents develop in separate bubbles until the year 1492, when everything suddenly connects. Verified contact would shatter that illusion of tidy separation. It would paint a picture of a world still largely defined by isolation, but one where that isolation was occasionally, miraculously, pierced. Human history becomes not a series of isolated acts, but a symphony played by many orchestras in separate halls, where every so often, a musician from one room would step into another, play a single, haunting note, and then slip away, leaving only an echo in the air and a strange new seed in the garden. This does not simplify our past, it deepens its mystery and grandeur. It asks us to hold two powerful truths at once, to marvel at the profound originality of cultures that grew from their own roots, and to wonder at the rare, shimmering threads that may have connected them across the deepest blues of our planet. The map of our deep past is not yet complete. This journey through the botanical evidence reveals a world both more isolated and more connected than we once believed. The strongest cases, like the sweet potato's voyage across the Pacific, point not to mass migrations or lost super-civilizations, but to limited, likely sporadic contacts. These were moments of meeting, perhaps even bidirectional exchanges, where a single plant could become a living souvenir of a journey that history forgot. The Atlantic whispers with far fewer clues, its storms guarding older secrets, while the Pacific hums with the stronger, though still debated, melody of the Kumara line. What this means for science is a vital lesson in humility. Our understanding of human migration is not a static monument, but a dynamic model, constantly updated as new tools let us read the fine print written in seeds and genes. Each carbon-dated tuber, each sequenced genome, is a chance to refine the story, to add a faint, tentative line to the map. This process honors the enduring human spirit that drives it all, a boundless curiosity that pushes us to build canoes against the horizon, to wonder what grows on a distant shore, and to never stop asking how our world was woven together. That same curiosity is what brings us here to explore these mysteries side by side. If you want to join the next investigation into history's greatest puzzles, be sure to subscribe.